那个于老师，刚才钟老师说就我介绍了。
OK，
那我们就开始吧。
OK。
好，各位老师、各位同学们，大家上午好，大家晚上好哦。于老师那边是早上，上午好，于老师。那我们今天呢，非常的荣幸邀请到了我们的于日清老师来给我们做报告。那我也非常的荣幸担任今天的报告的主持人。非常感谢钟老师和 EEH 的团队给我这个机会哈。那下面呢，我先按照这个程序要求，我就首先简单的介绍一下我们的于老师。那我们于老师目前是在德克萨斯大学泰勒分校的当副教授。然后我们的于老师在华中农业大学毕业，硕士毕业之后呢，先后在中科院水生生物所和香港大学和路易斯安那州立大学都获得了硕士学位，所以我们于老师拥有3个硕士学位。之后呢，是在罗格斯大学获得了博士学位。那本人也非常的荣幸，在美国罗格斯大学访问的时候，跟于老师是同一个实验室。但可惜的是，遗憾的是呢，我到罗格斯大学的时候，于老师已经毕业离开了。但是我们同一个实验室呢，经常听那个我们的老板陈马巴 K 经常提到我们于老师，一般都是表扬我们于老师在实验室工作多么的细致和努力，做了非常多的工作。之后博士期间的工作也是最后发表在《EEH》上面。所以之后我离开之后呢，那篇文章那个陈马巴 K 还特意发给我，也是非常大的贡献。于老师经过这么多年在汞的微生物循环方面获得了很多的成绩，成果也发表了很多。相信的接下来我们于老师的报告会给我们更详细的介绍。那接下来我们就请我们的于老师开始我们精彩报告吧。
OK 啊，谢谢。国内的同学们、老师们，晚上好。按照这个钟老师的建议。我们因为用英文来讲，所以我也是用英文准备，所以我下面就开始向你们报告我们的研究，好吧？这个好像有一个这个很奇怪的，OK。Okay, it's very glad to have the chance to show the study from our group. So I really appreciate this kind of opportunity and the platform. And what I'm going to introduce is the studies on microbial mercury methylation by using gene characterization methods. So firstly, you can see the kind of map showing the distribution of, I don't know how to get rid of this bar here. Okay, so the top 25 mercury emitting coal burning kind of power stations in the state. So then you can see even in the East Texas alone, which is where my campus is located, you can see I have 7 different power stations, so quite close to the campus. Then you can see here just list the top 25 the mercury emission in 2009. So the top one is Martin Lake. I have a picture here, so which is very close to our old campus. So just 1 hour's drive. So you can see we still are alive. So that means that we still need, I mean, in Texas, we need a lot of kind of electricity. And then you can see it's still burning coal because it's much cheaper than other like resource. And then we're doing kind of study recently. So then I have 2 graduate students taking samples and did the gene detection and then try to do mercury analysis here. So you can see also in this region have other 2 or 3 carbon pulse stations. So what I'm showing here is just give a sense, so the emission of the mercury in the state. Then also I found a graph. So I think from the paper from Operation National Lab. So it's showing the global distribution of the mercury emission in 2010. So you can see the countries have the highest mercury emission include China. So then India. Then follow that. So you can see have USA, so Russia, and also the top is Colombia. So somewhere here. So I really don't know what kind of data they base on it. So, but if you're interested, you can find this paper. So I think it's very interesting in showing the kind of overall mercury emission, right, globally, which is very interesting. So then here just showing the major mercury cycling procedures. So in the ecosystem, we just talk about the major like source, right? It comes from the coal burning electricity power station. So and also have other like industrial applications. So we so-called the mercury legacy. Right? So we stay out there for decades. So then you can see there's a kind of very unique feature for mercury. So especially inorganic mercury, like 0 and 2+. So once they emit from like a coal burning power station, it can be transferred by the atmospheric kind of environment movement. So then in that way, they can actually drop almost everywhere, right, on Earth. So that's actually why mercury contamination is so like ubiquitous. So then once the mercury drop into the land and finally we go to the aquatic ecosystem. So then we know there's also another feature, right? It's so-called particle reactive. So they can react with the particle and sink down to the sediment. So in this kind of anaerobic environment, so you can see a lot of kind of microbials include like sulfur-reduced bacteria, iron-reduced bacteria are shown here. So the methanogens, they can convert those are kind of less toxic mercury 2+ or 0, so into methylmercury, which is a kind of neurotoxin, right? So then we know methylmercury is a kind of highly lipophilic kind of compounds. It can be easily transferred and biomagnified through aquatic food chain, then accumulate in fish. So when people eat this kind of fish, then it can cause a kind of really health problem, especially it causes a kind of damage on brain. So we know methylmercury, they can pass through the so-called the brain-blood barrier, so reaching in brain. So then cause a kind of neural kind of toxicity. So especially also low IQ on newborn baby. So what we interesting is the this kind of procedure. So for the microbial, so-called the methylation, or you can say this is just a way biotic synthesize of methylmercury. So it's a 100% actually biotic, means microbial cause. So then here I have another cartoon showing the very complicated transformation of mercury. So we now have 3 different phases, right? Mercury 0, which is a water type, and 2+ and methylmercury. So we can see almost all the different steps are gene regulated. So it means you can see mercury 2 plus can be reduced to 0, right? So then you can see it's a kind of regulated by MeHg. So then we know mercury 2 can be transferred to methylmercury, then later on, so you can see 2013 gene was the mercury methylation gene was identified. So that's another big kind of focus. Then methylmercury can also be degraded. So the one like a gene is MeHg gene, which is very important. So in this kind of process. Just a little bit about the historic studies in this field, and I will go over the several case studies in our group. So, such as the contribution of coexistent sulfate or iron-reducing bacteria to mercury methylation, and the role of methanogen syntrophic in the microbial methylation. And we briefly introduce kind of the update studies in this kind of very unique genes. And if I have time, then I will go over a little bit about the challenges in methylmercury degradation. So you can say this kind of phenomenon, microbial methylation, was originally recognized by Jensen general. So in 1967, so they took some sediment from the lake. So then from in Sweden. So then they add the mercury 2 plus, then they can detect the synthesis of methylmercury. So then later on, you can see another group from UIUC. So they use one methanogen, methanobacteria strain MeHg. So they break the cell apart, then mix with mercury 2 plus, and they find out they detect the synthesis of mercury 2. So then they further did a kind of mechanism studies, then proposed the coenzyme method called biochemistry. So biochemistry, we know this is a kind of vitamin, right? B12. So they actually can help the transfer of methyl group, so from like a metabolic pathway, then to mercury 2 plus. So in that way, you can see they can synthesize methylmercury, which is very interesting, right? So because we know methanogen, they can produce a methane. So the way for them to produce methane, they actually use the methyl group, right? So this actually makes sense. But this kind of hypothesis was largely neglected. So after Richard Barsa, so from Rutgers, did a kind of very classic studies. So what he did is just use the so-called inhibitors. So use two inhibitors. One is for molybdenate. So you can see here, it's very similar to sulfate, right? So it's a very specific inhibitor. So to sulfate bacteria. In that way, just once you add molybdenate, the sulfate bacteria actually consider this as a sulfate. They can take up, but they cannot do any like a sulfate reduction. So that means that they actually cannot grow based on that. So that's the way actually inhibit. So then another one is 2-bromide ethane sulfonate. So which is here. So or SR. So this is actually another very unique kind of inhibitor to methanogens. So you can see once they add this here, so they took some sediment from the salt marsh in New Jersey. So this actually Richard Barsa group was in the same department. So with my PhD advisor. So he retired actually like 30 years ago. Then Tamaba came moving. So then you can see what I'm saying is here. You can see once they add this so you can see you can complete inhibit the methanogenesis or the synthesis of methane, right? You can see here. But once they add this kind of data here, they actually did not stop or inhibit the mercury methylation. They actually stimulate. So, but once they add molybdate, you can say completely stop the synthesis of methylmercury. So in that way, they're actually very straightforward. Right? The claim, the first time proposed the sulfur-laden bacteria are the major mercury methylators. So in that way, actually indirectly reject previous Wu's hypothesis about the methanogens. Then you can see there did actually a series of studies, actually very fundamental. So then you can see that one study is they use the isolate disulfovivin disulfuricans IRS. This is string actually was very important. Then they feed on this kind of string, the grow this string with two substrate. One is serine, another one is formate. So both were labeled by carbon-14. So then they grow this bacteria, and after days incubation, you can see they can detect the methylmercury, which actually contain the radioactivity. So they actually confirm the carbon here. So in this methyl group, in methylmercury, actually come from either the serine or formate. So then they also use a chemical, so propyl iodide. So once I add that, so you can see they cut off this kind of acetyl-CoA pathway. So also at the same time, they cut off the synthesis of methylmercury. So in that way, they actually can propose the mercury methylation pathway actually through acetyl-CoA pathway. So this is actually very important kind of theory in this field, especially for those kind of incomplete, so-called incomplete sulfur-laden bacteria like disulfovivin, disulfuricans. So later on, this string I was lost. So after he retired, so we could not find it. So that's just a kind of big lesson. If you have a very nice kind of isolate, yeah, you probably need to deposit it in different locations, right? So then in 2006, so two groups almost at the same time confirmed iron-laden bacteria actually are very important, another very important group of in mercury methylation. So you can see the group from UC Davis. So they use the similar kind of inhibition acid. So once they add molybdate, you can see they actually stop the sulfur reduction, right? Almost 100%. But it didn't inhibit the mercury methylation, which is here. You can see only in 40% inhibition. So that's just a kind of indirect way to confirm. So sulfur-laden bacteria actually are not the major one, right? Especially in the second setting. So then another group from Cindy Gamo, so Smithsonian. So they directly tested this kind of mercury methylation by using geobacter, several like iron-reducing bacteria, very classic species. So you can see once they added mercury 2+, almost like 14% can be could be converted into methylmercury. So that's actually very high, right? So you compare with other species. Okay, then we switched to the case study we did it before. So then you can see this one case study. I don't know how to get rid of this bar here. It's very annoying. Then you can see this is actually the project from DuPont company, because Virginia State and also one famous environment organization in USA. So we call Environmental Defense Fund. They sue DuPont company because they caused a really big contamination in South River. You can see there's a north part of Virginia. So the river, you can see it flowing from south to north. So the reason they cause a contamination because they have a kind of factory here to produce like an acetate fiber from 1929 to 1950. So by use mercury sulfate as a kind of catalyst. So it just causes a lot of leaking. So in the river sediment and also river bed. So then this actually is a very big project. So because they need to pay a lot of money, so millions of dollars. So only then they need to do a kind of 100 years by monitoring kind of program. So that involve I think more than 10 different universities. So then you can see we set up a lot of different 10 different assembly sites, so downstream from the factory, and then did a lot of kind of analysis. So you can see the river system actually is very interesting. So what we did actually is only focus on the microbial part. So then actually there was a lot of data. So we only pick some very interesting data in the 2 seasons we did the studies. So then you can see the total mercury, so and the Methylmercury, so in the sediment, you can see actually vary among different sites. So this is different sites, some sampling sites, and also seasoned. So if you compare with other like a classic mercury contamination sites like Carson River, so you can see quite similar, so a little bit higher. So if you compare with the Methylmercury, you can see it's almost 20 times higher than the concentration sediment from Minamata Bay, in Japan. It's actually first famous contamination sites. You can see this almost the size from South River, actually 20 times higher. So then you can see this is EPA standard, so 0.3 microgram per gram in fish. So to draw a line here, you can see almost all the fish found in the river has been contaminated with mercury. So some fish even contain 10 times higher, so kind of methylmercury in their muscle or tissue. So not only fish, you can see in aquatic and terrestrial birds, their blood also contains mercury too. So the reason for that is just those kind of birds feed on the fish, right, on the invertebrates in the river. So that's cause a kind of trophic transport. So what we did is, so you can see, we took the sediment. So I did a kind of quite similar kind of inhibition and stimulation test. So we add sulfate, because the sulfate is kind of like a electron receptor, right, to sulfate bacteria. So it's just like a full for those kind of bacteria. So you can see we only choose 3 sites. So then we measure the background, like sulfate concentration or ion concentration. Then based on that kind of level, we add different concentration of sulfate. So try to see what kind of response will occur in those kind of microcosm, right? So you can see once we add sulfate, the methylation rate increased 1.6 and 2.6 times. That's actually very significant. So once we add molybdenate at the 3 different sites, so you can see the inhibition by like 25 to 28%. Then for iron-reducing bacteria, actually, we don't have like a specific inhibitors, right? So that's we figure out one way. So try to add like a newly synthesized the free hydride or free hydroxide. So we know this is kind of newly synthesized actually quite bioavailable. So then also based on the background concentration. So once we add the free hydride, so then you can see actually I can stimulate, especially the lower level. So it can stimulate 50 to 100%. So kind of methylation rate. So this kind of study just first time indicate both iron or sulfate-reducing bacteria actually can play like a very important role in the river sediment, which actually very interesting kind of result. So in the same time, we also did the Methylmercury degradation tests by using kind of facility. So we actually spent several months to establish. So then now you can see we can track the gaseous products. We know the methylmercury, right? Once it degrades, the carbon here will be released, right? Either as a carbon dioxide or methane. So that's the kind of purpose. They try to trap the gaseous products. So you can see that the methylation rate actually is also varied among different sites and even different seasons. So then most interesting here is that you can see in the maize samples, we have the gaseous product, which was quite dominated by methane. So but in the August sample, you can see it was dominated by carbon dioxide. So you can see this actually the methylation, which is really highly specific. I mean, it can site specific and season specific. So at that time, because the mercury methylation genes have not been identified, right? So we are using the 16th RNA genes, then try to target those are live microbia. So we injected RNA, then convert into cDNA. So in that way, we can do the clone library and the sequence. So although this is not like a next generation sequencing, you can see we still found quite a classic kind of sulfur-laden bacteria here, and also the iron-laden bacteria. So which actually was quite lucky. Then the second case study is the mercury methylation or contamination in one forest lake. So in Adirondack, so which is kind of upstate of New York State. We know this region actually is highly isolated. So it's supposed to be quite clean. So, but still, we found the mercury contamination in almost all the different fish. So that's why we try to figure out what kind of mechanism happening over there. So this is kind of also a big NSF biocomplex project collaborated by several universities. So you can see we did a lot of kind of analysis in different locations. And finally, I actually found the flowing mat, so mainly composed of sphagnum, so then some grass. So also this is a whole the part of a stuff, I should say, kind of floating. There's no root, so no detection on the salmon, so which is very interesting. So this I should say kind of hotspot. So it means we found the highest methylmercury here. So then we try to understand why. We know the mercury methylating microbia actually almost 100%, right? It's kind of are kind of anaerobic microbia. So, but here actually is quite aerobic, right? We try to understand why this is really the case. So then, first of things, we tried to characterize the sulfur-laden bacteria. So then use one specific genes, so very unique to sulfur-laden bacteria called dissimilatory sulfide reductase, or SRB genes. So we're using D gene. So then you can see what we find actually most of the kind of species actually are very novel. So we cannot find any reference species in gene bank. So then not like what we thought. So for the classic 6 group are sulfur-laden bacteria, right? We found actually very few. So did find some very similar to the sort of bubbles. Then some clones quite similar to a Syntrophy. So then also we did a kind of enrichment of methanogens. So after like 70 days incubation, we did find some kind of methanogens, but we know it's very time consuming if you want to isolate methanogen. So we didn't do that. So we just use a quite simple way to confirm. So because we know all the methanogen contain very unique like a coenzyme, right, called F420. So if there's a kind of methanogen you put on a seaweed, you can emit fluorescence. So that's actually quite a way to determine if you have methanogen or not in the enrichment. So then we use the genes, which is kind of very unique for methanogens we call methyl coenzyme M reductase or MCR-A genes. So try to characterize this kind of methanogens. So again, you can see you find a lot of kind of species actually really kind of new. So could not find any like a reference species, similar species in gene bank. So we did find some species, clones, clonings, quite similar to like the methyl-spirulone ungated GF-1. So we know this is a very classic kind of methanogens, right? So that just gave us a very strong signal. So leading us to think about if the methanogen again can methylate mercury or not. So because we know Richard Bassel's studies actually largely rejected Wu's hypothesis, right? So then you can see we gathered the pure culture, the Hungarian species from Germany cultural collection. So then based on the media they recommend, because they actually contain like using like a sodium sulfide, okay, as a kind of reductant when we prepare media, right? So we need to add. So we know if you work on anaerobic like a culture, you know, actually it's very trivial, right, to do that. And then in order to get rid of the residue of oxygen, you need to add some reductants so to consume the oxygen. So we use that the recipe they recommend. So at the beginning, so you can see actually they didn't find any methylmercury synthesized. But later on, I realized the sulfur actually is a kind of inhibitor of methylation. So then we switched to a titanium chloride. So then you can see very obvious kind of mercury methylation occurring over the 5 days incubation. So then we use other methods, also other strains to do the measurement, okay, the production of methylmercury. So you can see this is the Africanus, which is a confirmed kind of mercury methylators. You can see actually quite higher than, but this actually, it really depends on what kind of media you're using. And then also we compare with other like species like Geobacter and also the supervivian ND132. So by using different substrate. So at least you can see the methanogen so can produce methylmercury kind of comparable kind of level with like a Geobacter. So we got this data just quite early, so in 2011, I think 2010 or 2011. So then we really have problem to explain it, because at that time, the methanogen has been considered as not kind of mercury methylators, right? So until the Oak Ridge National Lab group, they finally identified the mercury methylation genes in this two classic species, one ND132, PCA. So then you can see also the Hungarian species also contain the consensus motif, right? So the hgcAB, the protein. So this actually is very unique. So then we realized actually this is really what happened. So in methanogens. So for this kind of study, you can see really Jerry Park kind of paper published in Science 2013 really is a kind of big breakthrough in this field. So the reason for that is because previously we really had problem to have a kind of direct kind of link when we studied the mercury contamination, right? To how can we make a link to the function genes of microbes. So this actually really opened a window. So I know this the whole story actually is very complicated. So the study of genes actually lasts probably 20 or 30 years. It started from I don't know, probably Jody Wall. So then later his postdoc Elians. So then the team, the big team in National Oak Ridge National Lab, they spent a lot of time and money, but it really break through a big like a barrier in this field. So you can see this gene contain actually as so-called hgcAB genes contain two parts. So then you can see the purple part is hgcA. It seems to be a little bit larger, right? So then yellow is hgcB. So then the roles of the two parts are actually a little bit different. So from like Alex Mabak's paper, so it seems to give a better kind of explanation. So then you can see the hgcA part is more involved the transfer of a methyl group. So now you can see the transfer of the methyl group from the enzyme, right, by using an encoding enzyme, like methyltransferase. So then step by step, so transfer the methyl group combined with a mercury 2+, then to synthesize methylmercury. So during the process, you can see the coenzyme copper, so also get oxidizing. So you can see that in order to continue to do it, so we need to make sure this kind of copper 3 reduced to 1, right? So that's our kind of role for hgcB. And also it seems that hgcB involved the binding of mercury 2+ with the methyl group. So based on what did they find, we did a kind of the blasting of the gene, especially the protein, so hgcA and B. So then we found at least another 8 different methanogens contain this kind of the gene or protein. So then we stuck this tree, so you can see they're actually grouped together, right, for methanogens. So a little bit different from others. So then also we have a geobacter here and then SRB, so here and there. So in that way, this is actually quite early kind of people in this kind of methanogen. We propose methanogen really probably the third major groups doing the mercury methylation, which is actually I think pretty important because previously when we study like the environment in the field, we found actually we contradict the situation, right? You detect that like a sulfate actually is very low, but methylmercury is still unconditionally still very high. So then what was the reason? Right? So this is probably one reason, because without sulfate, methanogen or like an iron-reducing bacteria can do the mercury methylation. Right? So that means that the previous like a policy EPA try to control the emission of sulfate, right? Then actually it's probably not the best way to control the methylmercury contamination. Then later on, you can see Cindy Gilmour's labs extended this kind of confirmation to other methanogen species. You can see the green ones, so they confirmed other kind of species of methanogens, which is very interesting. So then we move to another like a case study. So because in the Adironic project, we found like the SRB, right, sovereign bacteria, and also a lot of kind of central bacteria, and also methanogens. Then also that led us to think about if the syntrophy can play some roles in the mercury methylation. So then, as we know, there's a lot of kind of so-called syntrophic relationship among microbials, right? And two classic one include like the disulfur-waveling with methanogens. So even without sulfate, right? We know disulfur-waveling, they actually can do fermentation. So they can ferment lactate or other product to produce hydrogen. So you can see the density here actually is quite small, the value, negative value. So it means that they cannot produce a lot of energy. But you can see once they associate with methanogen, the messenger actually can use the hydrogen here, take away the hydrogen, then to make methane. So then you can see the delta G here, the negative value actually is very high. So once they couple together, so then they actually can grow really well, even without sulfate. So this is what happened in those kind of wastewater treatment strategy. So then another one is the central vector. You can see here, you play the same role as the sulfur-waveling. They can do fermentation to produce hydrogen. Then a methanogen can take away the hydrogen producing methane. So you can see this here, actually that is a positive. So that means that you need energy to improve. So this kind of syntrophic relationship actually is very common in the natural environment, especially like as sediment. Right? So we simplify and try to test this kind of syntrophic relationship by using the several species. And here we use two kind of the self-limiting bacteria. So one is ND132, another one is Africanus. So then culture them individually and co-culture, so mixed with methanogens. So what you can find here is the Methylmercury production. You can see here, you can see the co-culture was stimulated 6 to 19 times higher. So compared with the single culture. So here is the same. So you can see once they have a co-culture. So that's actually a really big kind of increase, right? So then also we detect the growth of the bacteria. So you can see co-culture can produce more cells. So then also we tested another kind of association. So the methanogen with central vector. And also we can find the co-culture can stimulate the mercury methylation, but not other two species, two kind of association. What was quite interesting is just we found actually the kind of association between the sulfur-waveling and central vector. We know both of them can be considered as a self-limiting bacteria, right? This is actually waste sulfate. So you can see the co-culture also can stimulate the mercury methylation, so 2 or 3 times higher. So especially here, you can see that. So that's actually quite surprising. So the mechanism for this kind of new association, have not been reported previously. So we try to figure out what the reason for that. So we found some people indicated that the sulfur-waveling species actually have been considered as the most competitive self-limiting bacteria. So once they grow together, then the central vector, like a kind of self-reduction for this kind of species, will be inhibited. So then tend to be to act like a fermenter. So they can ferment propionate to produce hydrogen. Then the disulfur-waveling can use hydrogen to do a kind of sulfur reduction. So we know the sulfur-waveling can use a hydrogen, right? So which is very interesting. We can separate. So there's 2 cells. So once we grow them by using flow cytometer, so you can see they actually grow. The co-culture actually can stimulate the growth. So then after testing the different cultures and also co-cultures, we try to figure out what's a kind of fundamental mechanism here, right? Then we spend a lot of time trying to figure out the kind of relationship. So what kind of mechanism? So we calculate all the delta G for the different cultures by using different substrate and also the co-culture, right? Either in freshwater or saltwater systems. Then also measure the methylation rates. So in this kind of different cultures and co-culture. then plot this two set of data together. And then we found actually quite interesting kind of positive relationship between delta G and the methylation rates. So that just gave us a kind of another kind of sense. The delta G probably is a kind of somehow kind of determination factor, right? When we determine the methylation rates. Okay, then I move to the current kind of update studies. So we know after the hgcAB genes have been identified, so then you can see recent studies actually on the novel species which contain hgcA genes has been expanded very quickly. So you can see probably 100 different papers published. But it seems a lot of kind of papers or studies that really kind of focus on the next generation sequencing and try to extend the kind of the frontier of novel species of phylum which can do mercury methylations. But not many studies actually really confirm in the lab if they really methylate mercury or not. So still you can see from the Cindy Gilmore paper, it's a little bit old, but still I think it's very interesting. So as we know, the current perspective for mercury methylating microbes include sulfur-laden bacteria, right? It's kind of dominated by the sulfur-laden, and then iron-resistant bacteria, then methanogens. Right? From this paper, actually, they really did some kind of confirmation, at least in the new phylum, right? Firmicutes. So you can confirm several species. So this actually is really very interesting. So to help us know what really happened. Because in my view, the species contain hgcAB genes, probably not necessarily mean they are really kind of mercury methylator. So who knows? So but this is just my view. Then you can see this kind of review paper we just published last month. So we spent a lot of time trying to explore the novel species or phylum, so which actually can do, which contain, or can say contain this gene, hgcA genes. So then you can see it really kind of expand kind of knowledge. So from what we found here, so except this kind of blue highlight, so which are confirmed mercury methylators. So all other species actually not confirmed, but they contain the mercury methylation gene or protein. So this is a kind of protein tree. So then you can see it really pop up a lot of kind of new species. So then that really changed our perspective in this field. That's quite similar to iron-resistant bacteria. Here, not only in iron-resistant, but also include other species, the methanogens too. So you can see some species actually they never heard before, like that. Acidobacteria, which is a very new phylum, right? So they are actually very dominant in the soil. I know because when we did the random project, we found a lot of kind of acidobacteria, but just cannot identify because it's just so new. The reason for that is it's very difficult to isolate Q culture. So that's probably another reason. It's very difficult to confirm because without Q cultures, how can you confirm, right, in the lab? So if they can do methylation or not. So then switch to the last part. So I talk about the mercury degradation. Right? We know this actually is a very important field. So when we really want to do some kind of bioremediation. So in this field, you can see most studied aspect in this field actually probably is the mercury resisting genes, right, or operon, or the mercury operon. So which is most occurring aerobic microbes like ecology, Pseudomonas. These are really well studied. Then we know have two very important genes, right? MeHg, which are actually involved they are the kind of regulation of the reduction of mercury 2 plus, it didn't label here, to mercury 0. Right? So that in that way you can volatile, so evaporate from either your culture or site. So it can be kind of as a combined. But anyway, you just drop it somewhere else, right? Then another gene is the MeHg gene. So you can see you can degrade methylmercury into either it's mercury 2, mercury 0 or 2 plus. Then also can emit the gaseous products, right? We talk about it, so methane or carbon dioxide. So this actually still this review a little bit old, but still very useful. So and then also we did a kind of study. So on this isolated species, Pseudomonas puritae, was originally isolated from Brazil in the landfill site. So you can see this strain actually is very powerful. So once you add methylmercury, so it can be degraded as a kind of mercury 0. So within like 24 hours, almost like a 70%, 71% was gone. So very quickly. So, but we have problem to identify the MeHg gene. So the reason for that is just the MeHg genes are very variable. So among different species, even within one general species, I will show you the tree here later on. So then here's just have a new review paper. So from Tamabaki and Dr. Bawa Gu. So very nice review paper. So we propose the different pathway for methylmercury degradation or demethylation. So you can see I have like a biotic demethylation. Right? So like he proposed what kind of organisms here, the genes or mechanism here for the products. So you can see the one we just talked about is the so-called MER-dependent pathway. So all mercury-resistant pathway. So most you can see occurring in aerobic. So it means they need oxygen. Okay. And then it rely on MER-A, MER-B. I'm not sure the anaerobic, this actually quite new. So it seems to include some facultative anaerobic, even active. Then another one actually is not well studied. So most occurring in those kind of anaerobic microbial, like sulfur-living bacteria, methanogen, iodine bacteria seems to be new too. So the mechanism actually are unknown. So, but the products, the so-called reductive oxidative, based on the review we just proposed, is mostly depend on the metal, so the mercury, like phase, right? So if it's a mercury 2 plus, think about the mercury, right? That mercury over there can be considered as a mercury 1, right? So that's actually oxidized. The so-called reductive demethylation, you can see the mercury 1 plus to 0. So that's a kind of reduction, right? So then Dr. Gu recently published a paper and showing the methanotropes actually can also do kind of degradation, which is very, very interesting. So do the kind of mercury degradation. Then abiotic mostly kind of occurring in aerobic, like a water column. So for them, UV light, we know, right? They actually can degrade. So and also other chemicals. So then in that review paper, you can see the showing a tree, which is very interesting, but also give a sense. This actually is a MERB, like a tree, showing the within one species, like a bacillus, right? You see, they're actually not grouped together, which is quite weird, right? So the Pseudomonas is the same. You can see you have Pseudomonas here, and Pseudomonas here too, right? So that means that the MERB gene actually is really variable. So in that way, that actually we cannot design the primer to target. So especially for those kind of unknown species, right? If you have genome, Then if I have pure culture, then definitely you can do the genome sequencing. But still, you need a kind of conservative region, right, to identify it. So that's a kind of challenging. If you have other like a comment, probably, yeah, I'm glad to listen to you. Finally, I thank the teacher advisor and collaborators, like John Renfield, Charlie Driscoll, Mahans, and Dr. Bawagu, James Ben, and also my students, undergraduate students. Then the grant from NSF, so TCEQ, and also all university. Okay, I'm glad to take your question. So either you can ask directly or type it in the chat. Okay, thank you.
Thank you, Professor Yu, for your wonderful talk. And it's very impressive and very informative. I liked it. And now Professor Zhong just told me I should use English to make the introduction. So the question period. So I suggest, because most of the attendants are from China, so I suggest we can use Chinese, either English. And of course, you're welcome to ask questions in English or Chinese. So any questions? So if you have any questions, we can unmute your talk. Then I have a question. Yes, you talked about the syntrophy effect on mercury methylation. So, and then you confirmed it in laboratory experiment. Mm-hmm. How do you think the syntrophic effects on mercury demethylation? Do you think it is common? And
yeah, that's a good question. We have now confirmed. So I think that probably is highly likely. So well, I don't know. So that's a good point. Yeah.
Yeah, because we also observed the syntrophic effect in polypetylene. Mm-hmm. So, but we actually don't know how to confirm the demethylation effect. So yeah, I don't know if you have any suggestion about how to conduct this similar experiment, or for example by microcogen experiment or laboratory incubation. Do you think it is? Yeah.
Yes, likely. So I think see like what do we say in the slides from Bakay and Dr. Goose, the review people. It seems that they list all the different species. The problem is I see a lot of kind of syntrophic actually happening in those kind of anaerobic microbials, right? Then the mechanism of the degradation of mercury, which is it's not very clear. So, for example, we know sulfur-latent bacteria or methanogen, they actually can degrade methylmercury, right? But the mechanism actually is not really clear. So that makes the study probably quite difficult. But if you want to just use the culture, then definitely can do it, right? You just compare, use a stable acetate of methylmercury, so add it, so to compare the difference between like a pure culture and the co-culture. So that probably give us a kind of very straightforward kind of result, a difference, right? But in terms of the mechanism study, probably still very challenging. So, but it's a very good kind of view. So I think because for pseudomonas, I'm not quite sure for the anaerobic. So probably they actually not quite rely on the syntrophy. We know those kind of aerobic bacteria actually is quite straightforward. Like we find a lot of kind of pseudomonas that can do that. But it looks like it's more likely to happen in anaerobic microbials. The methanogen, I don't know. So that's something actually you can ask Dr. Gu. So that actually probably give. See, in the like a real environment, what I'm saying is the microbia actually not grow by themselves. Right? Always they have a kind of association with other species. So that's something I think syntrophy is a really kind of big topic. So when we deal with the real environment issues, so if you want to build a model, so you need to think about it just now. Okay, but two ES ions immediately before the methylation within the bacteria. Okay. Okay. This is a very, very interesting question. So I think there's several people reported. So like a mercury 2 plus, either they can the update process actually is quite interesting. So the other either we bind with like sulfide, right? So that now we can stimulate the uptake, or you can take up directly. So, but it really depends on what kind of bacteria you're talking about, or microbial you talk about. So then once it's take out in within the cells, then that's probably the way the process, mercury methylation happen. So I'm not sure I answered your question or not. Any other questions? Okay, there's one visit.
One more question.
Yeah, one more question.
Yes.
Okay, does mercury methylation and demethylation occur in human body? Okay, that's a good question. See, we have a colleague before actually. She published a paper. So in like the wastewater from dental clinic. So they actually found a lot of kind of. We know they in teeth, human teeth actually have a lot of kind of sublatent bacteria. So I'm not sure that's something I just very interested to know. Huh? So it's likely. So that's one. Another one is a lot of kind of studies showing invertebrates, right? So they in their digestion gut, they actually contain anaerobic microbia, actually include like at least methanogens, right? We know cows, the stomach actually contain a lot of methanogens. So, but it really depends. If they eat a lot of kind of mercury, right, take up, it definitely will happen. But usually, the toxicity probably will cause even more damage, right? Because of even the mercury 2 plus also can cause a kind of toxicity effect. So anyway.
Any questions? So it's a very good time. One more question.
Okay, sorry. Why does the methylation bacteria methylate mercury? Okay, if it is beneficial for the okay, that's a really, really good question. I think several like researchers proposed this kind of question. So like Jeffrey Schiffer, she published one paper actually talking about it. So the reason I think for bacteria to like a microbials to methylate mercury, one hypothesis is that once the methylate mercury, they actually can quickly release out. So that's actually a kind of protection mechanism for bacteria or archaea. So because it seems for microbials, mercury 2+ inorganic mercury or methylmercury, for them it seems shows not quite different toxicity effect. So that's one hypothesis. They actually can so-called facilitate. It's kind of speed up the release of the mercury from bacteria. So that's probably one hypothesis. Then another hypothesis is just we talk about the so-called the co-metabolic pathway. So that what I mean is just by accident, but it looks like later on you have the gene, right? So-called methylation genes. The benefit for that is just probably one is it can speed up the release of the toxic metals. So once it become like methylmercury, and another one is by accident. I'm not sure it can stimulate the growth. So usually it seems as not.
Any more questions?
Okay, that's one more.
One more.
If it means that the toxicity of mercury can be lower than, to be honest, I don't know. Huh? So the toxicity, what I'm saying is they mentioned it looks like it's quite similar to them, but I guess methylmercury is more toxic than mercury 2+. But it looks like it's better for them, easier for them to release out. So that's actually another study from Jeffrey's kind of paper. They found actually most bacteria, they can synthesize methylmercury, then they can right away release out. So from cells into the media. So that probably then if it's a mercury 2+, see we know a lot of microbia actually you can consider as a mercury as like other metals because they actually need other metals, right? If we pick up by accident, so that actually probably is more difficult for them to release out mercury 2+. But methylmercury seems that they can release out very quickly. Okay, another one. Okay, thank you. It's a very interesting question.
So if no more question, then we can read them. Thank you for your wonderful talk and your question period. You answered all the questions very good.
Okay, thank you. So I really appreciate you guys attendance. So and also this kind of platform. So I wish the journal can be more successful. Okay, later.
Yeah. Then, thank you.
Thank you.
Thank you. Thank you. And good night, everyone from China, and have a good day, Professor Yu.
Okay, thank you.
Yeah.
Recording stopped.
师兄，哎。